Next, we have a presentation from our senior field application scientist, Ian Villamagna. Today, his talk is titled Standardizing Scale-Up, Insights into Producing Clinical and Commercial RNA-LNP. Take it away, Ian.
Thanks, Joe. Appreciate it. And thanks everyone for joining us today. Really excited to be part of this. So today we wanted to kind of focus on sort of any sort of differences and changes that we've seen insofar as scaling up, looking at more of sort of what happens when we get to a clinical and commercial scale for these types of therapeutics. So we'll kind of walk you through what we see, some of the technology that we use in order to get to that point, and some of the challenges that we encounter. And we'll leave some time for Q&A at the end as well. So we'll be happy to answer any questions that may come from the group here. So it's really no secret that precision nanosystems and Cytiva as a whole have really gone deeply into this area of genomic medicine. And the real reason for that, beyond anything else, is simply that we saw the field going there as well. So we work very closely with drug developers at really a wide range of levels and areas. And I think we saw very, very closely and very quickly towards even before the pandemic that the field was really shifting to this area of genomic medicine. The promise of genomic medicine is really quite vast, right? So you have this ability to work with tons of different types of payloads that can do multiple different things: silence genes, express different genes, edit genes. And what we see as a technology provider in that space is just a lot of ability. So a lot of possibility for different types of therapeutics to be developed that can ultimately help patients much more quickly. What we, I think, are finding as we kind of continue down this pathway of really jumping into genomic medicine and doing this in partnership with drug developers is that we're in a space right now that is inherently complex. And this is something that I say all the time, and I know we talk about all the time. And I think still, though, sometimes we can undersell sort of the complexity of what's being done. So we moved from this space of drug development, where we were looking at small molecules with sort of single molecules and fairly simple synthetic pathways, into this space where we're now working with these drug products that are multiple types of excipients. You have an active pharmaceutical ingredient and nucleic acid that is inherently unstable. And so you've added all this complexity into this equation when it comes to making these drug products. The flip side of that, I would say, is sort of what you see on the right-hand side here. And that's the beginning of the possibility. So to go along with that inherent complexity, you have a higher range of possibilities that can be done with these types of therapeutics. So we've seen these types of therapeutics work from an in vivo fashion in a vaccine application. We're seeing them be expanded to different in vivo applications. And now we're seeing them even further expanded into more of an ex vivo application as well. And so with that increased complexity and increased work, we're seeing that really large possibility of what can be done. Within that sort of space, there are a lot of unknowns. And when we kind of look at the field now today, we still see delivery as a big unknown for much of the field. It's the area that we think is the part where we have to lend the most expertise. So the understanding of sort of what we have to do to take that active pharmaceutical ingredient, to take that nucleic acid, and encapsulate it in such a fashion that is going to adequately protect it, but then also have it function well enough once it gets inside of a body, is really what we're dealing with right now. The challenges within that space, within delivery space specifically, can really come down to a number of different things. The nucleic acid itself, the design of the nucleic acid, how stable that nucleic acid is, just the pure knowledge gap in the area. So it's not all that common to find people that have backgrounds in nanoparticles or in encapsulation of nucleic acids. One of the key challenges that we see still today with this type of development, and specific to the delivery of nucleic acids, does still come down to scale up. So scale up is not inherently straightforward. And if you kind of take a step back and think, again, back to something like small molecules, scaling up a synthetic process for other drugs, while still has challenges, typically is not quite as challenging as what we're dealing with today. But when we think about sort of the complexity of scaling up genomic medicines, so scaling up these drug products that have multiple excipients, we're really left with a place where it's not as straightforward as to just take what's being done at a preclinical scale and then running it in a larger batch to go to a clinical scale. I think if you talk to any number of us at Precision that come from this background where we've done this in the lab, scale up was always a challenge for us traditionally, and continues to be a challenge for our customers and for drug developers in this space down the line. Really, I think what it comes down to is that when you get to a higher scale of production for these types of therapeutics, you're met with new challenges. You're met with different challenges that just simply don't always show up at an earlier stage. So purely by using larger batches, you have additional questions of things like RNA stability from larger batches, lipid stability. Lipid formulation over a longer run looks very different than lipid formulation at a small run. The idea of process development changes quite a bit, right? So moving from something like an Amicon filter for downstream process into tangential flow filtration it's a very different process. It requires a lot of time and care to basically have a process that can be used effectively. And as we add all this sort of complexity from the size of what we're doing, you're increasing the variability within that process. So the likelihood that you're going to get something that is the same every single time does go down in that sort of scale up process. So this is complex, right? And this is something that our customers deal with on a daily basis, and we see the field dealing with on a daily basis. And as a technology provider in the field, we're cognizant that we're not the ones necessarily doing this work, right? We're not the ones going in day out trying to develop these drugs. But where we see ourselves come into play is really to really efficiently develop technologies that can lower those barriers that we're seeing in those labs for our customers. And so what we look to do with all of our technological platforms at the end of the day is reduce the inherent variability in what's being done. And this is something that anyone who's familiar with our technology will have seen at smaller scales, and now we'll see at clinical and commercial scales as well. So looking at something like a manufacturing platform, like our NanoAssemblr technology, which really takes a lot of that variability of production of nanoparticles out of the equation. So we've removed an area of variability at a preclinical scale, and now at a scale as well. So when we go to make larger batches, we've now removed some of that variability. We can take that same logic and apply it across the board for genomic medicines. So looking at things like lipids, if we have a really adequate lipid library that we can use, we're able to take different payloads and use different lipids and remove some of that variability, lower that barrier to entry for people that are developing these drugs and therapeutics. And so we talk a lot about our genomic medicine toolkit, which is on the screen here, which is our platform technologies that are utilized for the development of these genomic medicines. At the end of the day, what this really comes down to is leveraging the expertise that we have to remove as much variability from this process as possible, whether it's preclinical or into scale up, like we'll talk about today. We've been doing this for a number of years now. And so, I know that we do these webinars quite often, and we really do try as much as we can to really broaden the knowledge of the field as much as possible. And whether that's through regional webinars or regional seminars with local application scientists or sort of larger events like today, we feel it's very important that as part of being an end-to-end solution provider in the nanoparticle space, we are providing that knowledge and we are giving that knowledge back to the field. So making sure that what we're doing and the technology we're developing is not simply a standalone product, something that you can take into your lab, use, and then continue on with your day, but is part of a greater process where as part of using that technology, we're also lending that expertise. And together in that process, we're driving that development forward. And that's really what Precision Nanosystems always has looked to do. And as part of Cytiva, we're looking to do as well, is continue to develop those relationships with our customers and work together to drive that technology forward, to drive those drug developments forward. We'll talk a little bit more here about scale up, and we'll give sort of the scalable solutions for genomic medicines. But I think it is important to point out, really anytime we talk, that the technological solutions we have developed are incredibly versatile. And that's really important when you think about sort of the controlled release field as a whole, or the nanoparticle field as a whole, but also the genomic medicine field as a whole. So what we're finding very rapidly is that by having things such as a manufacturing platform that is really agnostic to what you're doing, so it works on a basic principle of bottom-up assembly, that gives you the ability to make different types of drug carriers, whether it's lipid nanoparticles or liposomes or polymeric nanoparticles. That ability doesn't change. And so we have a really wide range of what we can do with this technology. And what that enables essentially is everything else you're looking at here. So it enables a range of different drug products. It enables a range of different payloads, additional payloads like proteins and small molecules, for instance. And I think what it gives us the ability to do as we look towards the future of genomic medicine and just sort of where the field is going in general is that it gives us the ability to move very quickly and react to what's coming before it actually comes. So when you have technologies that are extremely versatile, the new sort of technology that's coming down the line in genomic medicine, we believe we're well suited to be able to respond to that and take that new technology, drop it into our platforms, and have it have an effect. So we're able to take these versatile solutions and really continue to grow on them, continue to expand, and continue to work with the field to develop new therapeutics down the line. So we'll talk a little bit more about sort of the scalable solutions for, specifically in this case, RNA-lipid nanoparticles today. And so we'll focus here at the start on manufacturing and kind of what that looks like and how that kind of functions from a scale-up perspective. And then we'll kind of close at the end here with sort of tying it back together. Again, as an end-to-end solution provider with precision nanosystems, we're not sort of a one-stop shop for manufacturing technology or just lipids or just instrumentation. We do really believe that the full sort of picture of what we do should be considered for the most effective solution possible. That being said, if we kind of take a step back and think about our manufacturing technology, our manufacturing systems are all based on the idea of bottom-up microfluidic technology at a small scale, and then bottom-up mixing as we get to a larger scale. So we are able to use this mixing to really precisely make nanoparticle populations that are able to be used in the body and are able to be used in a very homogeneous fashion, a very repeatable fashion for a number of different drug products like we talked about before. You'll hear us talk a lot about our NxGen architecture and our NxGen mixer, and this is really key to what we do. So what we have found essentially through a lot of the work that we've done and what the field of mixing has found through a lot of the work that they have done is that the most homogeneous nanoparticle preparations come from homogeneous mixing that's occurring. With these types of nanoparticle populations that we're making, we're leveraging a self-assembly process. So we're leveraging this bottom-up assembly. So you have all of your components dissolved into two fluid streams. Typically, in the case of a lipid nanoparticle, you would have your nucleic acid payload in an aqueous phase, your lipids in an organic solvent, usually ethanol. And we mix those two phases together to allow for that mixing to occur and allow for that self-assembly to occur. By having consistent mixing, what we're able to do is have consistent self-assembly of these particles. And what that gives us at the end of the day is a high level of control over the particle production process. So we're able to kind of dial in and have different sorts of attributes that we can pick up on. So we can do things like alter size. We can do things like add different sorts of lipids very easily. But what it also does is allows for a really high quality product. It allows for a really homogeneous particle population across the board. and gives us the ability to work in a space where we have a very high level of efficiency with our particles as well. And this is true whether it is lipid nanoparticles or other types of particles like you see on the screen here. The NxGen architecture is proprietary to precision nanosystems. And so you'll see this on our website a lot. You'll see us talk about it a lot, but you will see it in scientific publications as well. So it'll be referred to most commonly as a DVBM architecture. And what's really important about this architecture and sort of the reason why it enables everything it does comes down to how the mixing occurs. So unlike with other mixing platforms that rely on things like 3D structures to create the mixing, we don't have that with this architecture. And so what that has enabled us to do is take this architecture and be able to scale it, which allows us to scale the level of production of our particle population. This can be scaling up, but it can also be scaling down, which is really important. And so what it enables us to do is develop a manufacturing technology that is extremely flexible, that does have the ability to go through a scale-up process, but at any point in that scale-up process does have the ability to come back to a smaller preclinical scale where we can do work that makes more sense at that smaller scale and can really enable customers to move much more quickly through that drug development process. It's a small piece of architecture, and it's at face value seems seems like a small part of the process, but realistically what that architecture has done for precision and for sort of the field is enable everything you're seeing on the screen here. So that architecture enables us to have things like a manufacturing platform that can go all the way from a discovery stage, like we have with our Spark instrument, through to formulation development and formulation optimization with Ignite, into process development with Blaze, and then into clinical and commercial manufacture with our new commercial formulation system, which we'll talk a little bit more about in a second here. It also enables us to have a really good understanding of sort of how the production of these genomic medicines functions. And what that enables us to do is start to look at the development of things like reagents, look at the development of different lipids and different types of lipids, and increase the complexity of that development because we've reduced the variability from something like manufacture. And all of that is due to that microfluidic and the NxGen mixing architecture that you see us talk about very commonly. The other real portion where this all starts to fit and fits really importantly is within what sort of the Danaher platform is building. And so as part of Cytiva now, what we're really able to see very readily is how this all fits together with the other technologies in the portfolio that Cytiva and our other operating companies have as well, such as Aldevron or IDT. So what we believe we have developed here as part of the Danaher platform is the full ability to go all the way from sort of DNA or RNA payload, lipid selection, through to encapsulation, to downstream process, sterile filtration, and then fill finish. So really an end-to-end solution to develop these types of genomic medicines very quickly across the board. So today is mostly focused on sort of scale up and scaling up these genomic medicines. And so we'll kind of start to focus on that here a little bit more and talk about that in more length. Scale up is not something where it's a one size fits all sort of answer. And I think that's an important thing to talk about. And I think that we haven't always necessarily done the best job of showing that and saying that. The reason that I say that is I think sometimes when people see our instrumentation platform, like you see on the screen here, the logic is that Ignite is an early preclinical instrument, Blaze is late preclinical. So this is where all of process development and scale-up work is done. And then you just move into this sort of GMP space and you're able to make and manufacture medicines. Realistically, the scale-up process looks quite different. And what we believe it has to look like to be effective is to have all of these used in concert very effectively to make sure that we're doing the right work at the right scale. So we can do a lot of our early preclinical work, a lot of our formulation development work on Ignite, but we can use Ignite and Ignite Plus to begin process development work as well. So looking at things like increasing the speed of production of these nanoparticles or increasing the size, the volume that we're working with. We can use Blaze for things like large-scale process development. So looking at large batches and how TFF responds to a large batch of that genomic medicine or that nanoparticle population that we're making. And then finally, we can move into the clinical and commercial manufacture space with this commercial formulation system, which is new to our lineup here and replaces a sort of first generation GMP system that we had. And is able to work in line with everything else you see on the screen here. So functions very similarly. To kind of touch on this system that is brand new a little bit more, this system is wildly different than anything we have developed for a clinical and commercial manufacture space in genomic medicine previously. In my opinion, I think this system that we have developed is going to be the Ignite for the clinical and commercial manufacture space of genomic medicines. So Ignite brought this really intrinsic ease of use, this sort of comfort level to scientists everywhere in developing these types of genomic medicines from a very early scale. And what we typically see as we get to this manufacturing scale is a lot of that ease of use goes away, just by nature of the technology that we've been using. What we've done here with this commercial formulation system is really designed a system from the ground up, specifically for the production of genomic medicines and for the production of lipid nanoparticles. So what that enables us to do is take everything that we have done with our previous smaller systems like the Ignite and the Blaze and build on that, but keep that sort of ease of use that people come to know from precision nanosystems, which will streamline the process to get drugs into the clinic much more quickly. It'll be able to work in a variety of different settings and is a large upgrade on any current technology that we had previous to this. One thing that we did keep in place, and one thing we feel very strongly about, is the single-use flow path. This is going to increase the flexibility of what we offer, so it still enables single-use technology across the board. So enabling very quick, sort of rapid production of these therapeutics, but does so in a way that you now have this flow path that is in a single piece, meaning the sort of changeover for this flow path is very quick, it's very efficient, and it enables the work to be done much more quickly. So what we've really looked to do with this flow path and with the system as a whole is really, first and foremost, still produce those quality particles that we need to have to move into the clinic, but to do so in such a way that we're able to increase the speed, the efficiency, and the flexibility that we're working with to really drive these therapeutics into the clinic as quickly as possible. The architecture that we've talked about and the instrumentation lineup that we've talked about does scale based on this idea of the limit size principle. So this has been shown a number of different ways, really, with our technology. But what this essentially says is that the size of our nanoparticles we're making decreases as we increase the flow rate, increase the speed of mixing through that architecture up to a certain point. And then at that certain point, we reach sort of a plateau, which you can see on the screen here. As we go through and shift in terms of scale, what we find is that the flow rate in which we reach that plateau, the flow rate in which we reach that limit size increases with the scale of production. And so this is what we've shown here. First, starting with our NxGen mixer, which you'll find on your Ignite, for instance, into NxGen 500, which is our standard sort of mixer for Ignite Plus or Blaze or the commercial formulation system. And then into our new commercial cartridge as well, which has the ability to go to 48 liters per hour. We're able to make those same consistent particles across the board, but we're able to do so in such a way where we're increasing that flow rate. So we're increasing the speed of production to match what we're doing with the actual increase of production with the systems as well. And so we've shown this a number of times. We'll give some examples here as well in terms of scale up, and we'll use a lipid nanoparticle sort of application for this, an RNA lipid nanoparticle with a SARS-CoV-2 payload in it to look at sort of an example of how the scale up functions. The first thing that you'll notice essentially with these systems, and this is extremely consistent across the board, is consistent lipid nanoparticle conditions, consistent formulation conditions in terms of the size of these particles, like you see on the first screen, the PDI or the dispersity of those particle populations, and then the encapsulation efficiency as well. So regardless of whether we are working at an Ignite Plus scale, like you see in orange, or making batches that are 5, 10, or 50 liters, we see that the particle populations do remain the same across the board. This is also true in terms of looking at quality attributes across the board from different instrumentation as well. So in this case, what we've done is looked across the board with Ignite Plus and using those different mixing architectures. So NxGen 500, and then our commercial cartridge at 2 different scales, 12 liters per hour and 48 liters per hour. And then looking along the side here at, again, the size, the PDI, and the encapsulation efficiency that we get from this system. We feel very strongly that we have developed scalable systems where you can feel comfortable knowing that anything you're doing on the Ignite or the Ignite Plus does have the ability to be scaled up through the Blaze and into this commercial formulation system, and that the particles you're making at that preclinical scale are realistically the same as what you're going to make at a manufacturing scale. What that means realistically is that all sorts of work like this, like an animal study, for instance, that are being done, even if you're doing that at a preclinical scale, like on the Ignite Plus, it is relevant to what you're going to see at a clinical scale. It is relevant to what you're going to make for the batches that are going into patients. And so you can work with a level of comfort and a level of confidence, knowing that what's being done in a preclinical development does enable the same type of development at a clinical scale when you go to make your drug product. We've shown this in a number of different ways as well. So this is one example where we've shown in a rodent model and looking actually here for the IgG or the antibody levels from this therapeutic and looking and seeing that we get the same levels of response across the board, regardless of where they're made instrumentation-wise. And showing this again in terms of concentration, this time using an in vitro approach, looking at the differences that we see. And we don't see any main differences with the different scales of production here either. This is something that we've shown a number of times. There is more historic sort of EPO mRNA data. There's data from a more sort of robust program that we ran for a COVID vaccine on behalf of the Canadian government. And there's also data that our customers have published as well that's available and is able to be seen, where we see very similar trends across the board. So we feel very strongly that as part of this sort of full GMP production for RNA lipid nanoparticles, what we've been able to do in this portion, this NxGen mixing portion here, is take one portion of what we're doing here with this workflow and really take a lot of that variability out of the equation. What that is going to enable for our customers and for drug developers is to be able to spend more time on the parts of this process that are more challenging, such as sort of the initial stages. So working more at the Ignite on things like tuning aqueous buffers or tuning the drug substance, looking at different types of lipids, or for instance, process development. So looking at tangential flow filtration. If we can take as much of the variability out of the equation from the mixing process and know that we're going into TFF with particle populations every time that are consistent and are relevant to use, what that enables realistically is more rapid development of TFF and realistically easier development of TFF. Ultimately, what we're trying to do here is accelerate this timeline. So by removing variability and increasing the flexibility and the ease of use to any point in this timeline, we're able to basically take what we're doing here and accelerate that development to get those drugs into the clinic more quickly. So that's our sort of scale up in manufacturing. And I think for the purpose of today, it's important to still touch on sort of other technologies that really enable that complete solution as well. So the manufacturing technology and the ability to scale up these genomic medicines is incredibly important. But without the sort of other aspects of these formulations, without the other aspects of what we're doing here, you're kind of left with only one part of the equation. So we have spent a large amount of time recently working on our lipid platform. And so looking at different ionizable lipids, different combinations of lipids that can be used very effectively for different types of applications. So we have seen sort of the complexity to the lipid space right now, insofar as what happens from a litigation standpoint and what happens from a development standpoint with a number of our customers. And we began developing a sort of very robust library of ionizable lipids that we own the IP to, and that we're able to use either in off-the-shelf kits, like you'll find with something such as our Genvoy mix or our T-cell kit, or are able to license to our customers for use in the clinic if it's a lipid that is more specific to something they're doing. The way that we've really gone about setting up our lipid library is by taking the sort of 100 to 120 ionizable lipids in that library and then grouping them into areas that we know work quite effectively. So for instance, we have a subset of those lipids that we know work extremely well in a vaccine application. We have another subset that we know work very well in a gene therapy application. And then another subset that we know work very well in a cell therapy application. What we enable here by doing this and by starting to form these large overarching groups is really enabling the ability to narrow in and find more sort of complex areas based off that library. So as an example, if we look at something like cell therapy, what we have found is that within this ionizable lipid library, we have a number of different lipids that work very effectively to transfect T cells. And we've gone through and narrowed that down into a specific composition that is an off-the-shelf kit, so it does work quite well. But what we're able to do from there is continue to build on what we are looking at. And so what we're able to look at is using that expertise and continually developing new lipid mixes that have different types of applications from there. So we took what we learned from that T-cell application, from that cell therapy application, and continue to drive towards sort of developing different types of applications for it. And so this data that you're looking at here is coming from an upcoming product of ours that's going to be looking at HSPC editing in HSC cells. And so what we're able to do is really take sort of the development of these different lipid mixes and begin to narrow down from that broader library and give another sort of avenue to drug developers to work with solutions that work across all scales, across multiple types of technologies in the genomic medicine space. As you would expect, and this is important as well, these lipids scale with the platform, and that's something that that is really important to us. And when we're developing lipid mixes and looking for lipids to use as therapeutics and as sort of different modalities, we look at scale up as a really important and key part of that process. If we give you a lipid mix that works really well at a preclinical scale, but simply can't make it through the challenges of tangential flow filtration and into a drug product, we don't think we've essentially done anything there. So we do look very diligently at scale up and how these lipids scale through the platform. This is one example where we were able to take our T-cell lipid mix, so the T-cell kit, where we started actually at a discovery scale. So we actually started this on our smallest instrument, the Spark, and have shown through work in our labs that we are able to scale this up first into a more traditional preclinical model that uses the Ignite, and then further down the line, the ability to scale up into true process development and true clinical and commercial manufacture as well. The last sort of component of precision technology that is important for that sort of complete solution and is important for the scale-up journey is having a partner in that process. We do feel very strongly that the programs that are accelerated the most, the programs that get through this development as quickly as possible, are ones where we work very closely with the customer. We do have a full services department available in Vancouver, and what this really serves as is value added. So working with our customers at really any stage of their development process where they may need additional support. We come into this field with a wide range of expertise. I think you would be hard pressed to find another group in the world that has amassed this amount of people that have a background in the encapsulation, the nanoparticle space, the microfluidic mixing space, the lipid space, all kind of housed under one roof in Vancouver. What that enables us to do is take that expertise that we collectively have and disseminate it to our customers. And we do this through working in our services department as well. So we'll have customers that work with us at a preclinical level and do proof of concept work with us. We'll have customers that do simply analytical work with us, where we develop analytical methods on their behalf for their nanoparticles that they're making. We have customers that ask us to do the process development work on their behalf and get them ready to tech transfer to a CDMO. We look to be flexible here. The goal is to give as much of our expertise and knowledge to the field as possible, and to enable our customers to move as quickly as possible. And whatever that looks like, whatever it means, we're happy to have that conversation and work with your group to figure out what works for you in that sense. So just in closing today, we work very, very diligently. And I say we, knowing that I'm just the guy who talks on the internet, but our teams really across the world, Vancouver and beyond, work very diligently to develop technologies to really meet the needs of our customers and the needs of the field as early as possible. We do always try to look towards the future and look towards what's coming down the line in order to develop technologies that can answer questions and meet challenges head on as quickly as possible for our customers. We very strongly believe that any scientist, whether you're here today or anywhere that we work with in the world, can be a genomic medicine developer. And we think that with where the field is going and where genomic medicines are headed, we do believe that this is going to be the majority of medicine going forward. And that a number of scientists who don't come from that background, don't come from that genomic medicine space, will end up being genomic medicine developers. And we're here to help enable that journey and that pathway, both for the drug development process and through the learning process as well, for all of the scientists that are beginning that journey and working towards becoming genomic medicine developers. So you've seen a little bit about the scale-up process today. I know we had a panel this morning. We have a panel after this, which is full of great information with a little bit more insight into specifics, into scale up and some of the challenges and things like that. Please feel free to reach out to us. We're always happy to have a conversation and chat with you about your specific program or anything like that. But thank you very much for your attention.